Hi there, I'm Sarah Robinson from the Sainsbury Laboratory in Cambridge. I'm also a Royal Society University Research Fellow, and this is my Science on Sunday talk, coming to you from my house. I hope you're all doing well. I'm going to tell you a little bit about the work going on in my lab at the moment. So, people in my group are very excited about stretching plants. Here you can see part of a seedling, and we're stretching it. You can see the cells getting deformed. These cells are expressing a fluorescent protein so that you can see them when we look at them under a confocal. And you can see how we're stretching this whole piece of this plant here. And so the question you'll be asking is, why? Why are you stretching this plant? And we're stretching them in order to study how the plants grow. And why are we studying this? Growth is key to living things. Whether you're a plant, an animal, a person, a bacteria, growth is really important, and understanding it is one of the key questions for developmental biologists. It has implications in terms of how your hands are the same size, how tall you are, how big a tomato is, whether your crops are big and fall over or whether they're short and they don't fall over. And so there are applications for crops, and there's also just fundamental understanding about how growth is regulated. And for many of these fundamental questions, plants are actually one of the best ways to study it. Plants are good for studying these kind of questions for one reason, is that they come in all sorts of shapes and sizes. These are the two models that I'm using in my group. This is a tiny Marcantia, which is a liverwort, and you can see the scale bar here is 400 micrometers. So this whole thing is like a millimeter and a half big. And then I'm also using a Arabidopsis, which is sort of 20-30 centimeters tall. But if you look outside your window, you go on your daily walk, you'll see all kinds of plants around you. Really big ones, really small ones, different shapes, different sizes. And even if you just look within one plant, you will see a huge diversity. You'll see the tiny seeds that they produce, or these great trunks of a tree. You can see the flowers with their highly reproducible shapes. You can see leaves that might seem more variable in their shape and size. And this is really a great tool for people to study development and size regulation, because you get such diversity even within a single individual. And plants keep producing these organs while they're growing. So it's not like an animal where you're born with most of your organs. They can keep making them throughout their life, which would be quite useful for us at times as well. And so this tiny little seedling at the bottom here, this is what I look at. I look at the hypocotyl, which is this piece of stem that's present in the seedling. And I look at this because it has a very simple shape, so it's quite easy to study, but it's also very responsive to the environment. If you look here, this is one grown in the dark versus one grown in the light, and the difference in length is phenomenal. And it responds to differences in light intensity, in color, it responds to temperature, to all kinds of things. And this really makes it a very powerful tool for understanding size and growth regulation, because we can manipulate it so easily. We just shine a different light on it and we get a different response. So this is very helpful for us. And so, how do plants grow? Plants are made of cells. The cells have walls that connect them together, and the cells have salts and sugars and proteins inside them, and this draws water into the cell. And because the cells have walls, this enables them to withstand that pressure so they don't explode, and they can really build up this pressure inside them. The cell walls loosen so that the cells can expand, but this is done in a very controlled manner, so the cell doesn't explode, it just gets bigger. And plants can sense changes in this stress and reinforce themselves. in the direction of the stress, which again prevents them from exploding or ripping apart. They reinforce themselves with these really stiff fibers, and they align these fibers with the direction of main stress. So in this cell, if the fibers are aligned horizontally, then the cell will expand in the opposite direction because those fibers prevent it from expanding. And you can treat plants with drugs that interfere with the process of putting down these fibers, and then you can see the plant on the right, the cells are really bulging up, whereas the ones on the left are very narrow and thin and controlled. So you can see the role these fibers are playing in holding those cells in that shape and controlling the growth of the plant. And so, back to the question, why do we need to stretch these plants with a robot? We want to be able to measure the changes in cell wall properties that are happening during growth in order to understand them. And Hooke discovered the law of elasticity. He said, as the extension, so the force. And so he was relating the extension of a spring to the force that was applied to the spring. And so if you apply a force and you measure how much something extends, you can get an idea of its stiffness. And so this is what we're doing with the plants. We're applying a force to the plant and we're measuring how much they deform in order to get some insight into their mechanical properties. So how do we apply forces to these tiny plants? Well, in my group, we built a customized robotic device which we can attach to a microscope. And so this enables us to take those pictures that I showed you at the beginning, where we can see the individual cells. And the sample goes here underneath the objective of the microscope, and then this arm can move, and it can move a couple of nanometers at a time. And then we have a load cell here which can measure force, and so we can program this arm to pull the sample this way, and then we measure how much force is generated by that stretching. And these are connected in a feedback loop so that we can program it to stretch the plant until a target force is achieved. And all the while we're imaging the sample, and we keep it in water so that it stays alive. When we apply growth hormones, for example gibberellic acid, we see that the seedling can extend more than in the control environment where we did not apply this hormone. This hormone is actually quite interesting because it's the one that was targeted during the Green Revolution to make the crops smaller so that they wouldn't lodge. And because we're gathering images of the individual cells, we can actually see how some cells are much more elastic than other cells. So these red cells at the top, they stretch 10% when I applied the force to the sample. Whereas these cells at the bottom that are blue, they stretch between 0 and 2% when I applied the force. And so there's a gradient in the properties of these cells along the seedling, which means that the top part is responding more and growing to grow more in response to this hormone. In the control, all of the cells only stretched about 2 to 4%. So I've told you a little bit about how we can stretch these plants in order to look at the loosening of the cell wall and get some understanding about how this is regulated by growth hormones. But I also told you that the plants can sense stresses and that they reinforce themselves in a stress direction in order to get a precise growth in one direction and to prevent this big swelling up. And so this is something that we've also been looking at by applying forces that stretch or compress the plant, and then looking at what happens to those fibers that are being laid down to reinforce the sample. And what we found was that when we are stretching the plant, these fibers tend to align in the longitudinal direction, so they're resisting the stretching. However, if we apply a compressive force, then the fibers tend to be more aligned in the opposite direction. so they're resisting the force in the other direction, like this. And so you can see that in this sample, it would be easier for the plant to grow this way than in this sample where it's got these fibers. And so I used the robot to compress the samples and to look at the fiber orientation, and then I measured what was happening to their growth. And in a normal growth condition, the fibers are this way, and the growth is quite slow like this. However, in the samples that experienced a compressive force, here, the fibers oriented this way, and we see a burst in growth. So applying a compressive force to the sample actually led to an increase in growth, and we think that's because of this reorientation in the fibers, so that the fibers are no longer resisting elongation. You can imagine that this is helpful because the hypocotyl's primary job is to get out of the soil. So when you plant your seed, you put the seed in the soil, it germinates, and then it pushes through the soil. And so what I think is happening is that the seedling can sense that it's under the soil because of this force pressing on it. And this force is causing the fibers to change direction and causing an acceleration in growth, which lets the sample push through the soil. There's also contributions from the light and things like this as well feeding into it. So this is just one part of the pathway, but it's quite nice because it provides a mechanical signal to the plant that it's under the ground. And so I hope that I've shown you today that it can be very informative to stretch plants, that we can learn about their cell wall properties, which gives us some understanding of how the plants grow. And we can also learn about how plants respond to mechanical stresses, such as hitting an obstacle or pushing through the soil, and that they respond by changing these fibers. And so to acknowledge the people who helped me out with this work, it was mostly done in the team of Chris Kohlemeyer in Bern in Switzerland, where I was before I moved to the Sainsbury laboratory. I'd also like to thank my team here at the Sainsbury lab, where I just started my group, and also everybody in the modelling club and everybody else at SLCU for making it a great place to work and very welcoming. I hope to be back with them soon. And thanks to you for listening. If you have any questions, I think you can email me or contact me via Twitter, and I'll try and get back to you. Thanks a lot.